Hello, it's really good to be with you. Thank you so much for inviting me. I want to talk to you today about experience with biosimilar biologics in Europe. There's my declarations of interest, and I'm very grateful to Libs Pharmaceutica for allowing me to give this presentation to you. Today, I'm going to talk about a few things. Over the next 30 minutes, I'm going to start a little bit with therapy in rheumatoid arthritis. I'm a rheumatologist, and that's an important disease. I'm going to say a little bit about what biosimilars are and what they're not. I'm going to show you an example of data for a TNF-alpha biosimilar that's perhaps the most widely used in Europe, and then talk about guidance on biosimilars, real-world evidence, which is really important. And throughout my talk, I'm going to focus on adalimumab biosimilars, and in particular, the one that's the most widely used, Amjevita. But I want to tell you that I'm not advocating the use of any one particular drug. I'm choosing this drug because there's data on it, it's licensed and available in Europe, and it's the most widely used biosimilar for TNF-alpha inhibitors in Europe. I do use other biosimilars, and I also use biooriginators. First of all, I just want to share a little bit about myself. I'm professor of rheumatology, and here is a waiting room for my clinic. And you can see it's full of people. It's a busy clinic, and they're all complaining. Now, I hope they're not complaining about me, but they are complaining about their aches and pains. And as rheumatologists, we see a lot of people with aches and pains. There's a spectrum of problems. On the one hand, people with fibromyalgia. I don't know if you see people with fibromyalgia. I do see people with fibromyalgia. And when I see them, I try and help them out. I help them out of my clinic as fast as possible, because what I focus on are the more serious problems, such as rheumatoid arthritis. We know rheumatoid arthritis is a bad condition. If it's untreated, it doesn't only rot away joints, it can shorten lifespan. However, I must confess to you that as a rheumatologist, the tradition that we have is not good for managing rheumatoid arthritis, at least in the past, because many years ago, this was the treatment for rheumatoid arthritis. People would get admitted, given bed rest, and their joints put in splints. Not a great treatment for such a bad disease. And then, of course, because we had drugs that didn't really work, we had to rely on our colleagues, the orthopedic surgeons. And here's a typical surgeon that's saying, first, the good news, technically, the operation was a complete success. But as you can see, something has gone terribly wrong. Today, it's a failure on my part if my patients with rheumatoid arthritis need joint replacements. It's my job to stop surgery being needed. And then, of course, as rheumatologists, we started to develop drug treatments. And it says here, I'll give you something to ease the pain. I don't know about you, I would prefer the pain than the treatment. And whenever we think of drugs, we've got to be careful that we're not causing more problems by the treatment. And then, of course, today, rheumatology is very different. We're very cool. We have lots of treatments that are very effective, and we have drugs that can be very useful. We understand more about the disease, what's the science that's going on, and we know about inflammation. Therefore, with our colleagues in the pharmaceutical industry, we can develop new treatments. And this has been how we've developed the TNF-alpha inhibitors, which are the most widely used biologic for rheumatoid arthritis. Now, we all know that biologics are good, they're effective, they're fast-acting, they're pretty safe, but on the other hand, they're very expensive. And that has been a major problem in many countries. Good drugs, expensive, and therefore not available to most patients. So recently, we've been able to develop biosimilars. Now, biosimilars are drugs developed by very clever technology. It's effectively reverse engineering the biooriginator to copy it in a way that's clinically indistinguishable. And that's brought down the cost of biologics, and that's been a really major advance in our treatment of rheumatoid arthritis. But let me first tell you, on the left, we can see biosimilars. It looks the same. On the right, we can see other biologic drugs copying biologics, but these are not the same. And it's very important that we understand the difference between a biosimilar with very tight regulatory development and an intended copy where often there are shortcuts taken. In the rest of my talk, I'm focusing just on biosimilars. Now, here we can see two people. I know you might think it's the same person, but it's not. On the right, that's me. And on the left, you can see somebody copying me, and that's the actor George Clooney. Genetically, we are 99.9% identical, but we are two different people. Does that matter? Well, biosimilars are 99.9% identical to the biooriginator, but that doesn't make them identical. So we've got to be careful that what we're looking at are biosimilars that behave to all intents and purposes the same way as a biooriginator. And when I think about what data we need to be able to appreciate whether it's safe for our patients and it's right for us to prescribe a biosimilar, there are key questions. Number one, does it work? Number two, is it safe? And also, can we afford it? And the advantage of biosimilars is that it makes drugs much more affordable, and that's really important for our patients. In the European Union, and of course I must just say that England and the UK has left the European Union, but we're still in the same kind of system regulatory. And when we think about how Europe regulates biosimilars, you can see here in this diagram, this was accurate up till 2018. It's changed a little bit since then. But the thing to appreciate is there's a lot of drugs that are put forward to be approved of biosimilars. Some of them get discarded because they don't meet the strict criteria to be sufficiently close to be almost identical to the biooriginator. Some of them get withdrawn before the ruling. There are a lot of positive options, positive opinions from the EMA, but of the 39 that hold or held a current marketing authorization for approval, 3 of them were withdrawn by the companies post-approval. So this is somewhere where in Europe there's a very strict regulatory environment, and that gives me a lot of confidence as a physician that a biosimilar is going to be appropriate to be used in my patients. There's lots of studies, there's lots of drugs being developed. I've just summarized some of them. Big companies, Samsung Bioepis, Amgen, Sandoz. In Europe, these are the companies that have developed the biosimilars to TNF-alpha inhibitors. For the rest of this talk, I'm going to particularly focus on just one of them, which is Amjevita, and in its development, it had the code name ABP501. And I want to just show you this slide, which is important for a biosimilar. Frankly, it's the most boring slide in the world. To me, I'm interested in differences. In biosimilars, we want to have things looking similar. And you can see here a whole range of different assays, different attributes where everything is similar. Or if there is a difference, it's minor and that has no impact on the functional activity. A boring slide, but very important. This is what we want to see for a biosimilar. And then when we look at the early phase developments, we're looking here at the pharmacokinetics, and in white and grey, it's the Humira, the biooriginator adalimumab, either made in the US or made in Europe, and in orange, it's the biosimilar. I can't see any difference between the biosimilar and the biooriginator with the pharmacokinetics. Again, that's very important. But at the end of the day, I want to know clinically what goes on. And here we're looking at the clinical trials for the development, in this case, of Amjevita. And we can see here again in blue, we're looking at the bio-originator Humira, and in the orange, we're looking at the biosimilar Amjevita. As we look in these trials over 2 years, the developments of ACR 20, 50, and 70s, and that basically means patients with rheumatoid arthritis getting 20% better, 50% better, and 70% better. Again, I see no significant difference between the biosimilar and the bio-originator. This is all good, it's all very important, but as I'm going to show you a little bit later, this is not the only thing that we need to be careful about when we're thinking about biosimilars for our patients with rheumatoid arthritis. Focusing still on what goes on in Europe, the various European societies have offered guidance on how we approach biosimilars. At the end, I'm going to tell you about South America and North America, but now we're focusing on European societies. And if we look at EULAR, that's the European League Against Rheumatism, there are a number of publications with recommendations and guidance on biosimilars. And this is one of the first ones. I want to just draw your attention to the fact that in Europe, and particularly in the United Kingdom, we're really encouraged, if not enforced, to use the least expensive biologic. So for example, if I want to give adalimumab, I have to give the cheapest biosimilar to that. And the NHS has got big buying power. It will buy in block contracts for providing biosimilars. But on the other hand, we might start a patient on a biosimilar. We may swap once from the bio-originator to the biosimilar, but we will not swap any more. EULAR and the UK do not think it is sensible to swap between biosimilars. There's a variety of reasons for that, such as a worry about immunogenicity. But I highlight here that there's a one-time switch of therapy, and there's good data that that has no effect on efficacy and safety. And switching between different biosimilars is not recommended. There's a similar type of position statement from the European Crohn's and Colitis Organization. And again, it's fine to switch from the bio-originator to the biosimilar, but not other switching. And again, the advice is to go for the drug which has got the lowest acquisition cost. And that makes a lot of sense. And that's one of the reasons that the cost of the TNF inhibitors have gone down dramatically. And I believe that's a very good thing. I'm going to move on now to talk about real-world evidence. What I've been talking about in the slides earlier has been some data on the development of one of the biosimilars, and the data that I've shown you has been crucial to get the drug licensed and then approved. That's a really important thing, but when we think about it as physicians, we often want more evidence, particularly for safety and also for effectiveness of biosimilars. The randomized controlled trials that lead to the license are important to get the license, that's essential, but they have really lots of limitations. For example, I don't know about you, but I do a lot of clinical trials. When I do a clinical trial, the patient that goes in the trial doesn't necessarily represent the average patient in my clinic, because in trials, there are very strict inclusion and exclusion criteria. And they don't necessarily represent the real world population. They're also blinded. But when we look at drugs in the real world, that's what you're using and I'm using in the clinic, we get additional crucial information that complements the clinical trials. In the real world, we follow patients for several years. In the real world, we have diverse patient populations, just like our clinics. And in the real world, we can have additional outcomes that we measure, such as patient-reported outcome measures, PROMs, and economic outcomes. And it's possible that as time goes by, this type of data, or these type of data, can differentiate one biosimilar from another. At the moment, we're not there, but getting real world data is crucial. I'm going to talk you through now, for this next section of the talk, some clinical evidence in the real world for adalimumab reference products and biosimilars. So what's coming out within Europe in the real world experience of biosimilars to biooriginator adalimumab? Well, this slide just summarizes 4 studies. And we're looking at here patients in Italy, in UK, and other places treated with particularly Amjevita. This particular slide focuses on that. And as you can see on the right, as we go down all the column on the right, basically patients are doing well. In the real world, they're getting remission. In the real world, just like the trials, there's no change in disease activity when you swap from the biooriginator to the biosimilar. Now, this is a kind of a story that we get for all of the biosimilars because they have to be so close to the biooriginator to get the approval that these type of data in the real world are what we expect. And this is all very reassuring. These are relatively early studies, but as you can see, the bottom 2 studies are published in 2020, so they're pretty well up to date. And as we can see, as time goes by, this looks very reassuring, as we would hope. At this stage, I want to just make things a little bit more complicated. I've told you about biosimilars, in order to get licensed and approved, have to be so close to the biooriginator that there's no significant difference. There are some differences though, and that actually comes down to not necessarily the biosimilar, but the excipient or the solution that it's actually dissolved in. And here's a good example of something that's developed over the last couple of years. And this is just a report from a UK pharmacy document for the National Health Service. And this is looking at biosimilars, adalimumab, and injection site reactions. And we all know that when we inject drugs subcutaneously, some people can get an injection site reaction that can be transient, or it can be bad enough to stop them taking the drug. The bottom line is that the pharmacists actually summarized some evidence from a report that suggested citrate-free adalimumab was more tolerable than adalimumab where the drug has got it dissolved in citrate. It's hard to generalize this, they said, but there's a pointer that if you have a biosimilar without citrate, it might be better tolerated. So what do we see? Well, here's some more studies. Again, 4 more studies, all of them reporting in this year, in 2020. And again, what you can see is that switch failures, I'm looking at the top one, published by Rosenberg et al., switch failures more likely in people who swap to a citrate-containing biosimilar. So these are people on biooriginator Humira, they swap to a citrate-containing adalimumab, then they're more likely to have a problem and swap back again to the biooriginator. But when they swap to a citrate-free adalimumab biosimilar, that's less likely to happen. That's important, and I just mentioned that Amjevita, one of the many adalimumab biosimilars, does not have citrate in it. And of course, Humira has been reformulated to not have citrate, but these are some small things that can make a difference. But as you can see, across all the other studies, again, there's no significant change when patients swap from the reference drug to the biosimilar. And in the second study, this is from the Czech Republic, and we're looking at Samsung Bioepis biosimilar, similar other ones, but again, we're actually looking at very good results from people in the real world swapping from biooriginator to biosimilar. One further thing here, this is a UK study, and again, we're looking at people with rheumatological or dermatological or gastroenterological problems who require an drug. And the bottom line is, as you can see in these graphs here, we're looking at adalimumab 1, which is a citrate buffered biosimilar, and adalimumab 2, which is a citrate-free biosimilar. And in blue, we're looking at the proportion of patients who've swapped from the biooriginator to the biosimilar, but have to swap back again. This work because of an injection site reaction. And we can see here that that's happening, particularly, it's not the case in dermatology, which is interesting, but both the gastroenterology patients and the rheumatology patients who swap to a citrate containing, a citrate buffered adalimumab, a significant number have to swap back. But when we're looking at people who are swapped to a citrate free biosimilar, it's much less. And the most common reason for the switch failure was either injection site problem or a disease flare. And again, we're looking at, this looks fairly dramatic, but we're looking at about 14% of people. So it's not huge numbers. This is showing this UK study that switching is very effective when people are on biooriginator swapping to biosimilar, but there are some things that might mean it doesn't work so well. In this case, whether the citrate there or not can make a difference. Here we're looking at a different biosimilar here, SB5, the Samsung Bioepis biosimilar to adalimumab. Again, good results, very effective, looks just as good as the biosimilar, particularly no significant change in disease activity. The adalimumab trough levels were stable, and as I mentioned before, a few patients had to switch back again, and they're the ones that had the citrate, and that's what SB5 that formulation was dissolved in. So just a little bit of a signal, not the drug itself, this is just as good, but something that it's actually dissolved in that can cause a clinical minor, but sometimes significant problem. Another case series from Italy. Again, I'm just going through examples of real life data, of real life exposure of patients to biosimilars. Again, what we're seeing here, all of those slides are showing that there's very good response to people who are given, in this case, it's Amgevita again, single center, previously biologically naive, but some had received another biologic, and you can see here the patients get better. It's an effective drug, the biosimilar. This is people with psoriatic arthritis. There's a very low rate of adverse events, and this is confirming more the picture of biosimilar drugs being very effective in the real world. Now, at the end of the day, I mentioned earlier that what I like to see is a drug that works, a drug that's safe, and a drug that's affordable. Now, when we look at the retention rate, so how long are patients taking a drug before they stop it? Well, patients tend to stop taking drugs because it's not working, it's not safe, or they can't afford it. So it's very important to see data on drug retention rate for biosimilars. And here's a study, another one from Italy. We're looking at patients with rheumatoid, psoriatic arthritis, or axial spondyloarthritis who've had at least one course of the Humira, the reference adalimumab, or in this case, again, it's Amgevita, but we're seeing the same picture with other biosimilars. You can see here across the this graph, there's no real difference between patients who are on the reference drug or the biosimilar. They're all taking it, staying on it because it's effective and it's staying effective. These type of data are very important to reassure us that biosimilars are very effective, very safe, and appropriate drugs to be given to people. So just to summarize the switching studies I've been explaining, the real world evidence on biosimilar adalimumab is developing. It started off in Europe, where we're using a lot of biosimilars in small short-term studies, but as time goes by, the evidence is growing and it's very consistent. The drug that I've been particularly focusing on, because it's the most widely used, well tolerated, also works in people with inflammatory bowel disease. And one of the things that has been raised is the potential for something called a nocebo effect. So we all know about the placebo effect. The placebo effect, we expect a drug to help us. So even if we're on placebo, we might actually report some improvement. Well, the nocebo effect is the opposite. Patients may be told they're swapping from a Rolls-Royce expensive drug to a very cheap Fiat drug. And they might think, well, don't I deserve the expensive drug? Why are you swapping me to the cheap drug? Maybe it's not going to work. So the nocebo effect is something that we need to be aware of with biosimilars. Most of the data, pretty well all of it though, really suggests that biosimilars are highly effective drugs. But as I mentioned earlier, the formulation of biosimilars, not the drug itself, but the excipient, has got a potential to impact on clinical effectiveness. This may turn out to be a big issue. I suspect it won't be, because I think this is a very minor, nuanced change. But I think what's clear, there's no doubt that the data in Europe on using adalimumab biosimilars in rheumatological disease is very encouraging, very exciting, and the net reduction in cost is making a phenomenal difference as to how we can actually give good drugs to our patients. So just to summarize, the real-world evidence on biosimilar use in Europe, where we use a lot, there's a good history of biosimilars, is extensive and positive. It's important that when we talk about biosimilars, we think about drugs that are not significantly different from the biooriginator and that have gone through an appropriate clinical trials program. Intended copies I haven't talked about, but these can have a rapid program, not the same rigor, and are not guaranteed to be exactly the same. There is a place for intended copies, but I'm only talking about biosimilars. And in biosimilars in the real world, their results are consistent with the evidence from the randomized controlled trials, very encouraging. When we look at real-world adalimumab biosimilars, we can see similar effectiveness and safety, and we've now got some years of follow-up. Again, no new signals with long-term usage. The studies I've shown you have not been powered to detect any differences and don't have comparators. So though we can get reassurance about swapping from a biooriginator to a biosimilar, we as yet don't have data to say which biosimilar may be better than others, although the whole way that we develop biosimilars would make us suggest that actually we would not expect any differences between biosimilars. Real-world studies have showed variable discontinuation rates, and that could be due to the nocebo effect, which I've mentioned earlier. And of course, open label studies normally tend to have slightly higher discontinuation rates than blinded trials. I'd like to tell you that though the nocebo effect has the potential there, in the UK we tend not to see that. And I think the reason we don't see it is that we're very careful to educate and explain to patients about their disease, about their treatment, and why we might want to swap from a biosimilar from a biooriginator to a biosimilar. When patients understand this, they're very happy for it, and we always give them the option to stay on a biooriginator or to swap. Pretty well 99% or more will be happy to swap. And we also feel confident enough with biosimilars that we don't put all new patients on a biooriginator. Now most of our new patients, if not all, go straight on a biosimilar. We're confident they work, we're confident they're safe, and also the cost advantage of that means it's really a no-brainer that we should be using these drugs. So if you want to read any more, I'd recommend there's a nice there's a nice book, journal on biosimilars from work that's been going on in North and South America. And I was privileged to write the overall editorial for this. And it's really good that in the Americas, there's a development of consensus as to how you're approaching and how you're going to be using biosimilars. And I think this has been a really good thing for us as rheumatologists. Thank you very much.